Well, thank you all for coming to the 2018 OHDSI Symposium. Show of hands, how many of you are coming to an OHDSI Symposium after previously attending one of our prior events? All right. And how many of you, this is your first symposium? Okay. So for those of you who have been here before, those of you who are newbies, please keep up your hands. And for those of you who have been here before, I want you to turn and talk to somebody who's new here and tell them whether or not they're in for a good experience today. So please. So I don't know what you were just told, but I bet at least somebody told somebody that the OHDSI Symposium is not like most meetings, that we try to do good science, but we also like to have some fun, and we also like to throw a few curveballs every once in a while to keep things interesting. And so we appreciate that you've all come at 8 a.m. to start a very long day. You guys have the agenda, and the agenda says we're starting at 8. We're only a few minutes behind, so that's not too bad. And the agenda goes through that we are going to be collaborating together all the way through 5:30, and then we're going to be celebrating together with a network reception and Chan's wonderful music from 5:30 to 7:30. The agenda also says that George Hripcsak is going to kick this off, but I guess we just really weren't organized or something, because somehow George isn't going to be the first one to talk. Jenna Reps was very pushy, and she asked to actually kick off the OHDSI Symposium. So, Jenna, would you like to tell us why you're so anxious?
So I guess being pushy works. So my name's Jenna Reps, and I co-lead the patient level prediction work group with Peter Reinbeck. And I asked Patrick to have some time because we're at the point in our patient level prediction journey where we need help from you. But just to give you a background, the patient level prediction journey started in 2015. So this is when it was identified as a priority for research in OHDSI. And we started the work group in January 2016. So the work group was a bunch of collaborators from OHDSI who wanted to research patient level prediction and look at how we can actually improve the way that's done with observational data. And we meet regularly, and we're very open, so if there's anyone here who's interested in joining, we'd love to hear from you. And from the work groups, we've actually been working from 2016 to 2017 on developing a patient level prediction framework. So when we looked at the literature to see what was done for patient level prediction, we saw that there was a lot of issues. So a lot of publications were saying that papers that were being produced with prediction models, they often couldn't be reproduced. So they were lacking details a lot of the time, or there was some sort of error in the way they were actually doing the methodology. So we wanted to come up with a framework where you could have a very clear prediction problem. And we're going to do that from actually having set components in the prediction problem. And then we wanted to look at some of the proposed best practices in the literature, try and come up with a framework that could actually address those, so we could actually get models that would be transparent and reproducible. So we did a lot of work over those 2 years, and we created a patient level prediction package that had the standard classifiers for doing prediction, and it went end to end, so from the data that we have in the OMOP Common Data Model all the way to actually being able to get our models and validate it and see the performance of those models. So in 2017, we're at the point where actually we're able to release the software for that, and the software is very efficient, so even with the big data that we have available, you can develop the models very efficiently. And we also did our first PLP tutorial. So at the end of 2017, last year, we had a first OHDSI PLP tutorial, and we're going to continue with this tomorrow, so I'm sure I'll see some of you again tomorrow, which would be great, and we've got lots of fun planned for you. In 2018, we actually developed the toolkit more, so we added in the deep learning. So this is a big hype to see whether deep learning is going to improve prediction in healthcare. And we've added that into the toolkit, so this is going to be an exciting area for research in that methodology. And we also added in ensemble techniques. This is when you can combine lots of models together. And that, again, it's supposed to be better if you look at lots of different prediction problems. It should be more consistent. And then the third thing that we've added in this year is the network external validation. So there was actually a paper that was written that was looked at. If you develop a model and you publish this model, They were looking at someone else independently, so an independent researcher looking at this paper and replicating your model on a new data set and seeing how well it performs on their data set, this external validation. It often took 3 years or more for someone to do that. And often this was because it was hard to reproduce a model. It's hard, if you've got a different data set that's a different structure, it's hard to extract the data in the same way, and it's hard to actually see the process that you've done. But with our standardized framework and with our common data model, we can actually do external validation very efficiently. The software that we have makes it very easy for you to just share your model, and so you just have to point it to the new data, and you can do the external validation. And this has all been in our patient-level prediction version 3 release that we recently put onto GitHub, which is openly available to everyone. So we also had a publication at the end of the beginning of 2018 that was in JAMIA that details the standardized process that we have and talks about some tools that we've actually been developing. But we still need your help. So right now, we're at the point where we have the standardized framework. We have this way of coming up with models that are going to be transparent and reproducible. We have state-of-the-art software for this. So the software is an R package. It's very efficient for prediction. And we have the data. As a community, we have over 1 billion patients worth of records. So we're ticking all the boxes for those 3 things, and we're able to be at the situation now where we can start to do this large-scale prediction and explore the prediction landscape. But we're at the point where we need to know where we're going to focus. So there's no point doing a prediction model if you're not focusing on some useful public health area. So we need to know where we can actually do prediction that would make an impact to public health. So we need that from you, and we're hoping that you'll give us some ideas as to where we can actually focus our framework and our tools. So we said that we standardize the way we actually pose a question in prediction, and what that comes down to is there's 3 different components. So you've got your target population, and this is who you want to do your prediction for, and we'll also, most of the time we refer to this as T. An example of this would be patients with atrial fibrillation. And then we have our outcome, which we often call O, and this is what you want to predict. So this could be ischemic stroke. And then you have your time at risk, your TAR, and this is when do you want to predict the outcome occurring. Often this is relative to your target population index date. So this might be 1 day until 1 year after the initial atrial fibrillation diagnosis. But the most important thing is the clinical application. What is this going to why is this important to public health? Why do we want to focus on this prediction question? So in the example we just gave, it could be that you could identify who would benefit from an intervention of the anticoagulant therapy. So we need your help. You were sent an email around 6 p.m. Yesterday evening with a link to a Google Doc. So if you're able to get your phones and laptops out and head over to that Google Doc now, if you don't have the email available, there's a link here to go to. And also, if you have a QR reader on your phone, you can scan this QR code. So we want people who have the important prediction problems to start going onto this document now and start filling out the form. So in the form, you'll have the T, the O, the time at risk, and just a little justification of why this is clinically important. So I'm going to keep the link on the next slide, so you don't worry, it's still going to be there. So if you have your emails or you have this link, please go to there. But if you have ideas since seeing the email, you can start to put them on now. If you haven't thought of anything last Tuesday evening, and you can hear some examples that will hopefully inspire some ideas. So we actually thought there's 5 different types of prediction that you can focus on. So the more common one that you see published is the disease onset and progression. But there's also, you can use prediction for treatment choice. You can use it for treatment response, you can use it for treatment safety, and you can use it for treatment adherence. So we've got some examples here of the types of questions, the structure and the example. So in the structures, you'll see that T is generally in green, that O is in red, and time at risk is in purple. So it's the T, O, and time at risk that we need from you if you can think of a prediction problem that looks like this. So I'm just going to give people some time to go onto the Google Doc and actually have enter in and have a look at the Google Doc. The Google Doc also has this table as well, so you can read it on there if that's easier. So the link to the Google Doc, if you're manually typing it, is goo.gl/capital W, capital G, lowercase e, capital H, lowercase e, capital G. So if you have some time now to just have a look at that form and think of some ideas. It will actually... It will be open for about 30 minutes once I leave, so there's plenty of time. If you have more than one idea for a prediction, that's fantastic. You don't have to stop at one. We want to hear as many as you have. And we're going to be using this as a way to focus on the patient-level prediction in the future. So I see some people getting their phones out and hopefully getting some typing in. So this might be, if you know a prediction that you've been thinking about for a long time that could have some good intervention, this could be a good time to enter that in. See many people with laptops, hopefully going onto the link. So if there's anyone who's got an interest in a specific medical condition, it could be predicting that medical condition. And it could be in lots of different target populations. So you might want to look at certain age groups, certain genders, people with certain, taking certain drugs, or people with a certain condition. These are all different target populations you can have. So it might be if you do have an idea for one prediction, you could make it into an idea of multiple predictions, so just looking at different target populations. And the good thing is our framework is we're able to do this at scale. So if you have a prediction problem with lots of target populations and lots of outcomes, we can actually look at that and still come up with models very efficiently. Yeah, it looks like we're starting to get some entries right now. So we're going to have a little bit more on this page, and then, like I said, you've still got 30 minutes after when I leave to enter some more submissions. So if you think of any prediction ideas of the next 30 minutes, if anything inspires you, then please put them in. Which one? So no box for treatment. So like you should have like a T, an O, and a time at risk. So like it might be your treatments, your T. It's okay. So the form looks like this. So you've got some instructions about the different components of our patient prediction. And then you've got the table that I was showing, the different types of prediction problems. And then these are the forms where you can fill in. So this is your target populations. This is who you want to do the prediction for. This is your outcome, what it is you want to predict. And then this is your time at risk. So what would you? So is your treatment your target population? So is this who you want to do the prediction for? Oh. So if you were trying to predict. Yeah, so if you were trying to predict who was going to take treatment 1 or treatment 2, your target population would be both treatments, and then your outcome would be treatment 1. So that's how you, your target population is like everyone you want to do prediction for, and then the outcome will be what it is you want to predict. So if the treatment's for a given disease, yes, it would be people with the disease and treatment 1 or 2. And then it would be, the outcome would be the disease and treatment 1. So even if you... So if you have a disease as an outcome, you could specify lots of different target populations. So you could say, I want to look at it with people who have condition A, B, C, D. So you could say that here, I want to look at lots of different targets. And that's possible with our framework. Yeah. And then the last bit is why is this an important question? Okay, so if I pull up the form now, so... Responses? Oh, here. Okay, so we've got 23 responses now, so that's perfect. We're getting a lot of feedback, so that's excellent. Lots of work for us to work on in the patient level prediction group. So we can see some of the responses here. We've got people with having congestive heart failure. This is a very nicely defined T. We've got lots of outcomes, very nicely defined outcomes. And lots of good descriptions. So we're going to keep this form up at 25. This is expanding quite quickly now. So we're going to keep this up for another 30 minutes and hopefully get as many responses as possible. And as I mentioned, if you have more than one prediction problem, feel free to put it in. If you have more than T's for the same prediction problem, feel free to add that as well. The framework is very, we've made it large scale and standardized so we can do lots of T's and lots of O's. And that's not a problem at all. 26. This is fantastic. So thank you everyone for filling this in. This is very exciting. This is going to give us an area to focus on for the prediction in the work group now. So I'm going to head off now, but thank you for all this feedback. This is fantastic. And you've got 30 more minutes if you get any more questions.
Jenna's just so aggressive and pushy that she insisted to come up here. I figured that'd be worthwhile to give her a moment. So thank you, Jenna, for that. So now the man of the morning, George Hripcsak, will provide you a proper introduction to the community.
All right. Here we are. Do my traditional. Beautiful. So good morning, everyone. First, I always forget to say who I am, so now I put it on my slide. George Hripcsak. I am the director of the OHDSI Coordinating Center set at Columbia University. And so let me say welcome. Welcome to the community for coming and giving your time to this effort. We'll do individual thank yous later on, but thank you to the community for putting on the conference, both for organizing it, doing the work that we're showing, and paying for it. I won't wait on thanking the ones who paid for it, however. So Evident, Cleveland Institute for Computational Biology, Amazon Web Services, Deloitte, Janssen of Johnson & Johnson, and Bayer, all put in money and resources towards this conference, which we couldn't have. And we'd also like to thank the FDA CBER Biologics for their support of our symposium through their conference grant. So these things allow us to put on this conference for free, tutorials, which are mostly for free, and to have our breakfast burritos. OHDSI. So what I'm going to do for the beginning of this is talk a little for the people, because as we saw through the raise of hands, there are a number of people who are new to it. So I'm going to do a little bit of a review of what OHDSI is, and then go into some progress that we've had over the past year. And let me just say right now, your job is not to listen to me, but put in responses to Jenna's requests. So anyone who's looking down on their phone or working on their computer, I'll just assume that you are generating responses for Jenna. So thank you for that. The more people looking down and ignoring me, the happier I'll be. OHDSI is an open science community. Our mission, to improve health by empowering a community to collaboratively generate the evidence that promotes better health decisions and better care. So we're driven by the science and we're driven by the needs of healthcare and science. We're not primarily an infrastructure effort, not primarily a data model effort. We're primarily trying to generate evidence that improves care. Our values, which we set early on our creation, innovation, and reproducibility. We want to be innovative. We want to do it in a way that is believable, that it's about the community, and that's why we have this symposium and why we always strive to make it free. Collaboration within the community, openness. You achieve reproducibility in part by being open about what you've done, and beneficence, to make sure that what we do achieve gets distributed evenly across around the world, actually. Here is the world and how it's contributing to the community. I don't know if you noticed, but in the back corner over there is a map with QR codes for the various groups and their collaborators. And so that's a wonderful thing back there. When you go up for your snack or lunch later on, take a look and you can use the QR code to see each of the sites. But you see that we're expanding around the world. I will go, so this was not supposed to, I wanted to go quickly through the slide, so I will go, wait a minute, no, there we are. We've had, this is our 4th annual symposium, shown there in the bottom in red. We've had a lot of meetings now in our 4 years of existence. We have our first Europe meeting was this year, actually, and the second one is in the planning stages already, with a date soon being set. We had our first Asian symposium last year, and 2 symposia within China, China-specific symposia. And as I said, this is our 4th, and we have face-to-face meetings in the US. And we're getting to the point that this slide is no longer practical, and we'll have to go to a new format for next year. But the OHDSI effort is about working together, and so meeting in person is important, and that's why it's important for you that you guys came today. Of course, we can't meet all the time, so we have to do it on the internet, and we have weekly community web conferences. We have 15 work groups working on working to solve shared problems of interest. A couple of examples shown there. I'll show you the outcomes of some of those work groups later on. The forums.ohdsi.org. By default, when you join OHDSI, you become a member and you get all the messages come. So I retain all the messages that come in through the forums. I see, like you can see through your email, to keep tabs of what's going on. Of course, since we're a worldwide effort, it means that when you wake up in the morning, whether at 5:30, you see that someone's already been working hard and you already have 100 messages on your inbox. We have 2,000 users, have made 13,000 posts on over 2,000 topics in various areas, so we're very active. So a lot of work gets done over the internet. We're an open science effort, so it's not just open source. We do have everything we build is open source, but that's just a piece of it. It's open science, and a big part of that is sharing everything we do. Everything is out in the open. Both the problems that we choose to work on, and then how we work on those problems, what data we use, et cetera. Of course, the data that we use, patient-level data, can't be shared easily, but everything above that, in this pyramid, is made available so we can produce reproducible evidence. How does OHDSI work? Well, it's a federated database. You spend some time locally taking your data, converting it to the OHDSI data model, which is known as OMOP being the predecessor of OHDSI. That process, like if you've done it 100 times before, it takes a weekend, but if you've never done it before, it takes 2 months to do the first version, but you're not done yet. You then iterate and continually refine the data because you find both that you haven't done the conversion perfectly maybe, but the bigger problem is you learn that your own data aren't perfect and you have to go back to the source and see what's going on and why am I missing asthma in in 2005. Well, here's what went wrong. Those codes got misassigned or something. So that's a process that usually goes on. In fact, we still do it now 4 years later, for years, continually refining our data. Once you have your data in this common data model, we distribute the query. The query gets executed across the network voluntarily. The answers are aggregated centrally, and the group works on the results collaboratively, and everyone ends up a co-author on whatever paper is produced. We have 97 different databases. Now, there are many more databases around the world that use the OMOP data model, but as part of the OHDSI network, which I'm defining as having put your data, registered your database on the OHDSI website, there are 97 different databases, patient-level data, mostly health records and administrative claims data, but also hospital systems, clinical registries, health surveys, biobanks. Collectively, there are billions, literally billions of patient records. We're just short of 2 billion total records, but of course, Medicare has a patient and then Columbia has a patient. That may be the same patient, so there's a duplication. If you look at the data outside the US, it's 220 million, so I would say we're pretty close to 500 million actual unique patients in our database. We're about 500 million unique patients on our data network, and that's pretty good. So if you look along the top of this slide, you can see the components that go into making our infrastructure. There's the schema, the standardized common data model, then the vocabulary, then the conventions. I'll talk a little more about that in a moment, and then the analytics you put on top of that, and that's what we use to do our science. So going through each of them, here's our common data model shown here. We're up to version 6, announcing it as part of this conference. When you have a data model and you have a lot of users, you have 100 databases, there's a balance there. As we learn things about how to use the data model, we want to improve our data model. And as we pull in new types of data, we want to make additions to that data model. So that's one side. On the other side is, if you have 100 databases, it's a lot of work to make changes to those 100 databases. You want people to keep up so that when we spread the query around the world, everyone can run it and have it run identically. So we try to move at a measured pace, and how do we do that? Well, there's no we do it, you do it, through the CDM work group. So the community decides the pace. And there are individuals who are on the lookout for that. Of course, there are also individuals on the lookout for making sure we move quickly enough. And so this move from version 6 to version 7 is largely, there's a few new tables like the survey conduct table seen over there. But I don't know if I have a point. Largely, it's an evolution and not a revolution in version 6 going from version 5. I don't think the changes from version 5 for sites will be very hard. The vocabulary is very important. So now you have your schema. You have to put things into the schema. That's where the vocabulary comes in. So the vocabulary group that actually implements this, that is the community members and the OHDSI team, working with actually the National Library of Medicine and NIH to put together these mappings of, say, 80 or more than 80 now vocabularies from around the world and mapping to our standard vocabularies, which are largely SNOMED, ARCS, NORM, LOINC, and then specific vocabularies depending on the area. That's an ongoing effort, and if you're on the forums, you see that occasionally we hear things and we try to improve them, and we continually work to organize the vocabulary. Right now, working on getting the procedures into good shape in terms of hierarchy there. A lot of common data model efforts and vocabulary and ontology efforts skip this, it's implicit, we make it explicit, which is standardized conventions, what the Themis group has been working so hard on recently. That is, once you have a schema, and I remember this from the 80s working on HL7, you had the HL7 standard, and so that was the miracle, and we were going to put our data into HL7 and transfer it from our vendor system, say ancillary systems, to an electronic health record, and we were done. And then when we did that, we found that everyone interpreted the standard slightly differently, and no two vendors, even if they were sending the exact same data, did it exactly the same way. So no matter how detailed you are in your data schema, no matter how good your vocabulary, you're going to see differences. The Themis group is working on those differences and getting into detail. How do you interpret the standard? How should those data be put in? When there are questions, where are the answers? And that's what that group puts together. So that's a huge knowledge base being put together to help people do it. What's the method right now? Before the Themis group was the forums. You went on the forums. How do you guys store, death when this happens? And so now you can go and there'll be documentations. You can look at it as the wiki page for the forum. And then the Odyssey tools, I won't go through all of those in detail today. We have done that in previous symposia. This is the R Studio, and also there's the Atlas tool. I would like to point out the Amazon Web Services created the tutorial environment for our symposium this year so that everyone had free access to a large computing environment and can carry out the tutorial exercises, so that was wonderful. What we do with this infrastructure is to generate evidence, and we show here the three things we always talk about at the top is clinical characterization, that's counting, or tallying. So it's what do we know to be true, at least to the extent that we can know from our databases. And that's an easy one because we don't have a lot of assumptions there other than that the database is representative. On the right is shown as population level effect estimation. That's where we get to causality. Does the drug, say, or was some intervention, say a drug, on average cause an effect in some target population that we're interested in? And on the left is patient level prediction. At the individual level, that is for precision medicine, will this happen to me? What's the risk for me? So we always do those three things, and you heard a little bit about patient level prediction exercise that we're carrying out as part of this symposium from Jenna. The common data model and more of the Odyssey, in fact, not just the common data model, but the tools, et cetera, are being incorporated into several efforts. The top one there, All of Us Research Program, formerly known as the President's Precision Medicine Program, getting 1 million patients to donate their medical records and their specimen to carry out research. So that is based on the OMOP Common Data Model. And several Odyssey members are part of the data core for that whole effort. And we've asked all the academic medical centers that are part of that program to convert their data to the OMOP Common Data Model and send that centrally rather than just dumping a bunch of data at our doorstep that we have to then sort out. The good news about that is that since they have to put in the common data model, they have to do the work, they have the source systems. If they can't get into the common data model, they can figure out what went wrong in a much better way than I could do it once I get all the data centrally. The U.S. Food and Drug Administration, Biologics, is using the Odyssey initiative as part of their observational, and we're going to hear about that more this afternoon, so I'll get to that on the agenda, and you see that the conference grant was a large part of why you were able to come here for free today. The eMERGE network, which is a U.S. NIH-sponsored project, brings together health records and genomics using the OMOP Odyssey Common Data Model to make it easier to do phenotyping, because we find that we're in our 10th, 12th year now of this, and every time we generate a new phenotype, 10 sites have to implement that phenotype on their own data model. It's a huge burden, and what we're trying to do is make it more like Odyssey, where once we define the phenotype, you can just run it by pushing a button. Now, it's not that easy. The world is always hard. When we go to natural language processing, we don't have that solved yet, but for the structured data, we can actually push a button. The National Cancer Institute funding Odyssey to look at how the feasibility of using Odyssey to carry out cancer research, and the Innovative Medicines Initiative in Europe using the Odyssey OMOP data model as the basis of a pan-Europe database. So we're going to look at the Odyssey community in action and look at some of the things that were published over the past year. So breaking them into three groups roughly. First on data, Uli Prokosch's group, Miracum, Medical Informatics and Research and Care in University Medicine, which is exploiting the OMOP data model as part of a German data network. We're realistic, they're actually using the OMOP data model, they're also using I2B2, and they're using a third thing and putting together to implement this network, but as part of their application, they did a wonderful pilot project using Odyssey to push that forward. So that was a nice study that was published in the past year. Clinical data warehouse based on OMOP and I2B2 for the Austrian health claims data. This is a OMOP-based clinical data warehouse, which then uses Sean Murphy's new tool set for I2B2, which is the I2B2 analytic tools which sit on the OMOP data model. So that if you have an OMOP database, you can not just use the Odyssey tools, you can also use the I2B2 tools, and that's what they're exploiting. So it's nice that to come together of the two different common data model initiatives. From Columbia, expanding transplant outcomes research opportunities through the use of a common data model. And this is moving beyond the EHR and the claims data, using OMOP to expand a traditional transplant registry with new forms of data, new data sources. Moving now to methods, second group. So here, design and implementation of a standardized framework to generate and evaluate patient-level prediction models using observational healthcare data by Regina, who you just heard from. But this is a framework for scaling up the development of prediction models. Certainly there are a lot of prediction models out there, and every startup company in the world is creating prediction models for precision medicine. We're trying to do it in an organized fashion, in a way that scales up, that has the tools, and that does it in a reproducible way. Because how do you test? When you're making predictions on individual patients, it's even harder to prove that you were right. And so we're trying to do it emphasizing reproducibility. Empirical confidence interval calibration for population-level effect estimation studies and observational healthcare data. So this is the detailed description and evaluation of the way that we do OHDSI confidence interval calibration, which is the basis of the evidence that we presented last year and will be presenting this morning. And this is published in PNAS, which is a top-ranked journal, so it's giving visibility to the methods and the fact that it was accepted, showing the acceptance of the methods that we're using. This particular method is looking at how we handle residual confounding. Evaluating large-scale propensity score performance through real-world and synthetic data experiments, also by the group, which is the detailed description of OHDSI propensity score method and comparing it to an existing method used, I guess, over several decades, and showing its advantages over the previous one. So as I said, actually I should have put these slides the opposite order. This one is doing correcting for confounding, and the second one, this previous slide I showed, is the residual confounding that this one doesn't take care of. Published in Philosophical Transactions A, the oldest journal, science journal in the world. Not that many people read it today, as many as some things like the New England Journal. We're still proud to be publishing, I don't know, with Newton and Leibniz and Schumi, but improving reproducibility by using high-throughput observational studies with empirical calibration. That was actually the topic of last year's symposium, but it's basically the methods. So the previous two papers were the methods pieces. This is the full range of methods from start to finish of how you do reproducible research. In this case, in depression, I put under methods because although it's about depression, we didn't focus on the depression outcomes. And last, paper on vocabulary mapping. When people hear about OMOP, they hear they're going to map their data to a standard vocabulary, and they're always scared to death. And we always reassure them, number one, that you're going to keep your source data. So there's a column for the source field, and then there's a column for what you translate it to. So your ICD-9 and ICD-10 codes get translated to SNOMED. By doing that, we can do studies around the world and not just in places that use ICD-9 CM or ICD-10 CM or READ codes or whatever other codes from around the world. What this paper showed is that when you do that translation, you can achieve a trivial error rate. If you have a knowledge engineer create your SNOMED concept set for the phenotype, the error rate is on the order of, the max error rate was on the order of 0.2%. So error rates in electronic health records are in the 2% range or the 10% range, or for some things, the 50% range. 0.2% is a very tiny error rate, and that's what we were able to achieve across 9 phenotypes from the eMERGE network. Okay, now getting to some clinical studies. Comprehensive comparison of monotherapies for psychiatric hospitalization risk and bipolar disorders, Christoph being an OHDSI member. So these bipolar disorder treatment efficacy did vary by as much as 2 times in their study, which is shown in that graph. Also found that monotherapy was ended or changed within 2 months, showing that the short success rate of doing a monotherapy using observational evidence. Database studies of treatment-resistant depression should take into account adequate dosing. A huge percent, not quite 50%, but not that far from 50% of patients never achieved the minimum therapeutic dose for whatever medication they took, which means they're being switched off their medication before they even got the dose needed to achieve an effect. So that has to be taken into account in studies. The association of hemoglobin A1C levels with use of sulfonylureas, dipeptidyl peptidase 4 inhibitors, and thiazo. I practiced this ahead of time and it didn't help. Led in the diones in patients with type 2 diabetes treated with metformin. So this was a study of efficacy and safety for second-line treatment of type 2 diabetes mellitus. Remember, a couple of years ago at the symposium, we presented how people treat diabetes, and it's mostly with metformin around the world, 75%. But what is it for, and if you look at the guidelines, it says start with metformin, not a lot else what to do after that. So we were starting to look at that. We actually found only small differences among the second-line treatments, and in fact, that supports the fact that the guidelines are not that specific on which one to do, although there's further evidence now on that. So comparative effectiveness of CANA, I practiced it at a time, but I'm just going to call it CANA, SGLT2 inhibitors from non-SGLT2 inhibitors, and the risk of hospitalization for heart failure and amputation in patients with type 2 diabetes mellitus, a real-world meta-analysis of 4 observational databases. So this one took the sodium glucose cotransporter 2 inhibitors showing reduced hospitalization for heart failure and no increased knee amputation, which is something that had been shown as a possibility on a previous study. No increased risk versus non-SGLT2 inhibitors, and no differences among those of the SGLT2 inhibitors. The nice thing about this is all the data, all the results from this are posted on the OHDSI website. So again, moving to this model, everything. So it'll be in the publication, but in fact the data, not the data, the results are all openly on the internet. And not a publication, but a publication on the web. Remember last year we presented our incidence, that is, for every drug on the world market, for every side effect in SNOMED, what is the rate? This is not causal, this is the absolute risk, not the attributable risk, therefore it's feasible from a computational point of view. But the point is that we published in the sense of putting it on the internet. So if you just go to howoften.org and type in any drug, use the generic name because the user interface we haven't really fixed yet, any drug in the world, doesn't have to be US, and any side effect in SNOMED, so there's 300,000 concepts or diseases in SNOMED or something, it'll tell you the rate for a number of databases, and we're expanding that now to a larger number of databases. But again, about the publication and openness of OHDSI. So now, we're not just publishing papers within the OHDSI community, people are published from outside the OHDSI community are also publishing, so I'll just summarize them. I got these via Google Scholar, basically searching on OHDSI or OMOP, but there are a lot of other uses of the acronym OMOP, so I actually hand went through them to get rid of those. So clinical studies, there were 29 published since January 2018, so not even since the last symposium, but since January. And I won't expect you to read them, and I won't read them to you. For methods papers, there are 47 of them, so that's how many occurred in the last, say, 9 months. And about the data model, 137, so a lot of interest in the OHDSI and OMOP common data model. And if you get the slides, those are actually the papers, so that is not fake script. Those are actually the papers listed on the slide. So if you download it, you can get it. But you can just go get it off of Google Scholar. Many of them beyond the Latin alphabet, just to point out that OHDSI is a global network. And then just to finish up, a few comments about the community. My typical offhand comments. So one thing that I love about OHDSI and the OHDSI community is, you go and give a talk somewhere and you do it, and if people like it, they ask, and even if they don't like it, they ask, can we post your slides? Maybe on the second time around, people get something out of it. And usually my answer always used to be, well, let me look over the deck and I'll get back to you, or let me look at the lunchtime, I'll take a look and I'll get you back the slides. I'm going to delete a few slides that have new stuff and I'll send it back to you. Or, listen, I just got to email my colleagues and ask them if I can give you the slide deck, as if you've never been asked the question before because you don't really, At OHDSI, it's always go ahead, do whatever you want because it's already on the internet, there's nothing to hide. So literally, it's like you're the one, so you're in a panel of 4 people and it's like, I'll check on it, I'll check it on it, check on it. Yeah, put it on the internet right now, do whatever you want. So I like the openness of OHDSI. And it's a different way of doing research, and it's surprising to people that join OHDSI. This is the most bizarre slide, so I don't know how to explain it. These non-traditional, so when you grow up and if you're interested in science, you fantasize about, inventing something, and you read about Apple started in a garage, or Skunk Works, which was, Lockheed Martin, Skunk Works became Skunk Works, where it's a group of people come together non-traditionally and solve some problem. And it's about the group coming together, motivated not by being yelled at by your manager or not trying to get tenure, but there's some goal that you're working to achieve. And that, in fact, the expertise for that thing is there, but they're doing something else because they're so busy trying to get tenure. They're writing papers or getting grants and doing less research than they could do. And so the Odyssey community is an opportunity for all those people to, on their off hours, when they're not busy doing their job, to actually get science done. And this group coming together to do this. And to see that happen within the Odyssey community and all these things, and like every year we'll present these things, that it feels like a Skunk Works project where people are in their garage. Of course, we don't need the garage because we don't have too many technical things we need. We don't have to build the computers anymore. Coming together and do this. And that's like one of the funnest things about Odyssey. It's like living out your dream of when you were like 10 years old and fantasizing about what science was. The infrastructure of Odyssey allows this kind of thing to happen. So it's kind of like we've achieved a critical mass now. The data network with a consistent data model, with many, many patient records, so you can do things that couldn't be done before. The tools, in eMERGE, I mentioned the eMERGE project, and working with other sites working on the phenotype. We're on the phone and we're just working on Atlas, sitting on the odyssey.org website. I'm saying, here, try this, now you try that. And so there's a critical mass where we can just work together on stuff, and it's just sitting out there, and anyone else who wants to listen in on us could actually just go on the Odyssey website that second and go on Atlas and see what we're working on. But mostly it's about the culture of openness and sharing, and just if anyone raises their hand, they're in the group, and we work on stuff together. And the fact that when I just tell my guys, when anyone posts a question on the internet, Columbia's executing it. So whatever study you want to run, we're just going to be running it, period. And it makes it easy for us, the fact that we have a common data model. But it's odd for some people with a potential collaborator coming in. They're used to a hierarchy, and so, well, if I join you, then aren't I the senior author because I'm the senior most person who would be on the project if I join? And the answer is it's a group project. So first of all, just realize anything you tell me is going on the internet. Number two, we don't know where your name's going to end up. Somewhere in the middle is what I'd assume. The group somehow, and I don't know who the group is, will decide the order based on what best suits that project. And so it's a different way of doing work. So your name will be on the list, but we don't know where. So my last slide in this direction is that we have a lot of work. There's so much to be done. We need new groups working on things with great ideas. And so we invite people to come, and it's asking the question, I'd love to be in charge of a large famous study. So we invite you to do that. But I caution you, it's hard work. So the joke, I don't know if you've heard this before, is like, who in this room would like to make $1 million? And everyone raises their hand. Yeah, I'd like to make $1 million. You say, I didn't ask you how many people want to spend $1 million. I asked how many people want to make $1 million. How many people want to spend like 10 years of their life working night and day to pull something together that actually makes $1 million? And that's what it is. I'd love to run a large famous study. Who wants to spend night and day for some number of years to create that study? So this invitation comes with a caution. And I bring this up because of the group that will be presenting this morning. Here's the agenda. After the welcome, often what we do in the symposium in the morning is we highlight a particular project, the plenary session. I mean, like, I don't want to set expectations. Like, I just can't believe this is our best project ever. I mean, this is unbelievable, literally unbelievable. It's a change in the way, like, it literally is asking the world to change the way they do research and showing the evidence that we're right, that they should change the way they do research. I mean, it just kind of answers the question. So just like, if you're paying attention, that's a good one to pay attention to. Then we open up to the community, the Odyssey Collaborator Showcase. So you have all the stuff. This is where we come together. It's a very popular part of our symposium every year. So first the showcase, then the lightning talks, which were popular last year, and then come back for part 3 of the showcase. Then we open up to the community. We want to discuss this time the FDA BEST program. I mentioned that earlier, so we'll go over that detail in a panel. and finally close with, because this is a global initiative, the global progress and perspectives from the Odyssey community. That is the penultimate part. Then it's not the penultimate, it's the penultimate part. We have our closing session, which traditionally has been fun. I wouldn't be surprised if it was fun this year again. And then we end with our networking reception. It is not a networking reception. It is the last part of the symposium. It's when we give out our awards, and also we have our performance by Chan. You do not want to miss this part either. So this is an integral part of the symposium. Welcome to Odyssey.org, or welcome back to Odyssey.org. And I now hand it back over to back over to Patrick.
Thank you, George. That was a great introduction. So, let's see, I'm going to go on here. So that Jenna Reps, she just won't stop. She asked to be the main person. She comes up and gives the main talk, and then as soon as we step out the room, she asks, Patrick, I want back on stage. Well, I have a hard time saying no to Jenna. So Jenna, if you want to come back, you can have a couple more minutes.
So I'm invading the stage again. So we've actually went through some of the responses. Overall, we got 45 responses for different prediction problems. So thank you very much for all of that. That's great. In 30 minutes, lots of predictions for us to focus on should keep us going for quite some time in the work group. We actually picked 4 of these now, and we're going to actually show you these 4 that were picked, and we're going to ask for a live vote. So once this loads up, you're going to get a link to a website you can go to, and then we're going to see which of these 4 will win, and this will be the one that we're focused on the most. So this will be our priority within the work group. So you actually sent an email shortly ago, which will also have the link on it. So if you're able to go to your emails, you should be able to click the link. If you prefer, you could actually go to the link once it is put up on the form. And then we're going to see who wins out of the 4 different prediction questions that we've filtered through. All the questions were fantastic, and we've got plenty of work ahead of us, so much that we now need to prioritize. So the link is going to be another Google form. So people who actually did it manually before, it'd be a very similar process. But also, if you've got your emails available, you can go to the link that's sent. So the link for the voting is goo.gl/forms/N juicy 7 W Y Z Y 8 S lowercase b lowercase p lowercase b lowercase r lowercase h and 1. So I'm just going to give you
Check your email.
Or check your email. It's a rather long kind of bit at the end. So I'm just going to give you some time to get onto this before we start voting. Hopefully everyone's on there now. Just hang on a sec. So I hope you're all prepared now for the voting.
Why don't you read the questions? Why don't you just read the questions?
I don't have them.
They're right here.
So we have 4 questions. We got amongst people who have major surgery, which patients will go on to have deep vein thrombosis. We've got amongst within 2 days after the surgery, and then amongst patients with oral type 2 diabetes drugs, which patients will develop chronic kidney disease within 1 year. And then we've got the third question is amongst patients who have type 2 diabetes and are treated with metformin, which patients will develop diabetic retinopathy in the next 5 years following exposure start. And then amongst patients who have ischemic stroke, who were treated with anticoagulants, which patients experience a hemorrhagic stroke in the next 30 days after treatment. So these are the 4 questions that we're going to be voting on. We've already got 113 responses. So this is actually, thanks for the quick response rate. So we're up to 118 now. How many people do we have?
There's 470 people here.
So we've got about a third of the people at the moment. We're waiting for the other 2/3 to cast their vote on which one is the most important. So far, the one that is leading is patients who have ischemic stroke. So that seems to be at 31%, although it's closely followed by people who have the major surgery. So if you want the major surgery, you need to start voting now, or if you want the ischemic stroke to continue, you need to keep that. It's actually pretty close between those. We're still getting a few more votes for. Actually, yeah, people seem to be focusing on those quite a bit now. So it looks like the yellow's coming up now. The orange yellow is starting to follow. Could be a late winner if you want this one. So, green is taking the lead now, 37%, with almost half of you voting. So we give it a few more minutes for the voting, or another minute or so for the voting. Looks like green is getting quite a lead now. So if you do want the other ones, you really need to start voting. Up to 37.6% now, with 190 votes.
Jenna, can you make your screen bigger? We're blind back here. It's Kristin, not me. But Kristin and Kristin.
So hopefully you can see now. So the leader at the moment is the green with 37.5. So this is the ischemic stroke target population who are treated with the anticoagulant. So we're up to 194 responses now, so about 50% of the room. So we'll give it another 10 seconds. So if you want to vote, you've got 10 seconds remaining. Any final votes? Okay, 200 responses, and I think we're gonna finish there. So we got 37%. So the winner is the green question. So the people who have the ischemic stroke. So can I ask the person who proposed this question if they can stand up and introduce themselves and tell us why this is clinically important? We're going to have someone come over to you with a microphone. So if you can raise your hand and stand up.
Right up here?
So if you could,
Up in the front.
so if you'd like to stand up and just introduce yourself, and then.
Wait, I got you a mic. It's coming.
I'm very nervous about this. I'm from China. My name is Wang Qiu in Chinese, and call me QG for simple. It's my question. I thank you all of that. All of you, thank you that.
Well, congratulations for the most important question that was picked by the audience. Round of applause.
Because I have a very good friend, he's a neurosurgeon. He is very interested in this, and I'm already very interested in this because in the clinical practice, there's also very lot of people, a lot of patients get cerebral infection, and they will get some anticoagulations, and then sometimes they get a very high risk to after they have warfarin or dabigatran or aspirin or clobigrin. So I want to define or find if this person should take this drug or not. Yes, that's the reason from the very clinical practice. Yeah.
Well, thank you for your question.
Well, thank you all for humoring us to collect some questions. We know that one of the things we've realized in the community is we've got so many impressive researchers and impressive community members that we could accomplish an awful lot so long as we prioritize ourselves on the right problems. And I think across many of our different work groups, we've experienced the problem of having cool technology, but not necessarily knowing what questions to focus on. And so we thought, since we've got a captive audience, we'd capture from you guys what questions are important, because as George laid out, as a community, we need to prioritize our own community activities and figure out what questions to solve and what questions could potentially matter. And so I think that this will help us for the next year to think about what we could do for prediction. And hopefully, maybe next year we'll be able to show you some cool results of that question, or maybe some others. So thank you for all that, for that.